Hi guys, let's do section 2, mitosis and cytokinesis. Here are the essential questions. What are the events of each stage of mitosis, and what is the process of cytokinesis? Here are the vocabulary terms. If you haven't written these in your notebook, go ahead and do so now. And let's just dive right in. Mitosis is the process that makes new cells. Okay, during mitosis, the cells replicated genetic material. Remember that replication happened during interphase, during the S phase. Okay, this mitosis is the process of separating that genetic material. Okay, so during mitosis, the cell's replicated genetic material separates and the cell prepares to split into two cells. The key activity of mitosis is the accurate separation of the cell's replicated DNA. So we got to divide it exactly equally between the two new daughter cells. Otherwise, we end up with cells that have either too much DNA or not enough, and we don't want that. Mitosis increases the number of cells in a multicellular organism as it grows, and it replaces damaged cells. So if you've ever had a cut or a scrape, the cells that were damaged were replaced through this process of mitosis. Healthy cells were able to divide into two to replace the cells that were damaged. The first part of mitosis is known as prophase. Prophase is the first and longest stage. It is when the cell's chromatin, which is the genetic material, tightens up and condenses into structures called chromosomes. Chromosomes are made up of two sister chromatids, which are structures that contain identical copies of DNA. So that copy was made during the S phase, and those two sister chromatids are attached at the centromere. So those two sister chromatids attached at the centromere make up a chromosome. Here we have a picture of prophase. Okay, it starts with the chromatin condensing into chromosomes. The nuclear envelope breaks down, and something known as the spindle apparatus starts to form, which contains spindle fibers, which are the long, spidery looking things on your slide, centrioles, which are the brown things on that picture, and aster fibers. Okay, the spindle apparatus, its job is to attach to the centromere of each chromosome and move it around. Okay, make sure that it moves in the proper way. So the spindle apparatus attaches to each of the sister chromatids before cell division happens. Metaphase is the second stage of mitosis, and it's the shortest phase. This is when the sister chromatids, when the chromosomes are lined up directly in the middle of the cell. The spindle apparatus will pull and tug and maneuver until those chromosomes are exactly in the middle of the cell. Okay, that is sometimes known as the metaphase plate or the equatorial plate or the equator. Okay, really it just means that the chromosomes are lined up smack dab in the middle. After that is anaphase, when the sister chromatids pull apart. The spindle fibers made up of microtubules, they start to shorten, like reeling in a fishing pole. Okay, and this causes the chromatids to separate and move to opposite sides of the cell, to opposite poles. After that is telophase. This is when the chromosomes, the sister chromatids, the individual ones, arrive at the poles and they begin to uncoil. They are less condensed. Okay, a new nuclear membrane, nuclear envelope begins to form around that chromatin, and the spindle apparatus starts to break down because it's not needed anymore. It did its job. After telophase, cytokinesis really gets its stride. Okay, this is when the cytoplasm is actually going to divide. After the two daughter nuclei have formed, cells undergo cytokinesis. In animal cells, microfilaments, similar to the cytoskeleton, they'll start to form in the middle and they will constrict and pinch the cytoplasm into two, kind of like dividing one really big piece of Play-Doh into two smaller pieces of Play-Doh. In a plant cell, they can't pinch in half the same way because they have that cell wall. So the Golgi body will secrete vesicles and line them up in the middle, forming a structure called the cell plate. And that cell plate will eventually thicken into a new cell wall, separating the two cells. Okay, so plant cells have to do it a little bit differently because they have that cell wall that won't let them pinch in the same way. And that's it, guys. If you have any questions, let me know.